I based my PhD around working with the PPB genotypes that all of you breeders have worked so hard to create. And so this is just one project out of a whole program in my PhD. And so the other part of my program is looking at phosphorus use efficiency between farmer lines. But for the purposes of this call, I decided to present the data that I have on the yield stability analysis of the different genotypes across different organic conditions. So we'll get started. There we go. So for those of you who are not familiar, the concept of yield stability derives from the importance of needing broad adaptation to genotypes to many different environments. And so a lot of times when breeders have lots of different genotypes to test, they'll test them across many, many different kinds of environments. And so there's these different models that breeders use to look at how stable a line is. And so, you know, if we have on here is the performance, so whether that be seed mass or protein or yield, and then you have two different environments and you have two different genotypes, sometimes depending on the environment, the genotypes will react differently. And so, you know, we've got these different models, which is fine, but the one that breeders are most interested in is this crossover interaction. And so this is important because, you know, if you didn't have a crossover interaction, then a breeder would just have, you know, 6 different genotypes, test them, and then whatever was the best overall, we know that that genotype should be grown everywhere. But that never happens. Genotypes respond differently to different environments. And so to try to capture what is actually going on there, we try to do some different yield stability analysis to understand what genotypes do well where and what genotypes actually don't respond to better environments or what genotypes are more adapted to more stressful environments. So the objective of this study was to determine the broad adaptation of the PPB genotypes to identify stable and high yielding genotypes under different organic conditions across the prairies. Now, this is done across the prairies all the time for registration processes. A genotype that is being put forward for registration actually needs to show very good yield stability, very good broad adaptation, and very good yields to be put forward for registration. The challenge with the current system is that all of those yield trials are under conventional conditions. And, you know, I don't need to explain what the difference is to this group between organic and conventional, but there's just not enough data out there for that is derived from organic conditions across many different environments. And on top of that, it was a great opportunity to take PPB lines from many different geographic regions and test them across many different environments. And so we tested 25 farmer lines and 6 checks, and we tested them over 5 organically managed sites over 3 years. And so we tested at University of Alberta, the Organic Research Station, Organic Farmer Oxbow, Ian Cushion, the Parkland Crop Diversification Center, Organic Land in Roblin, Manitoba, an organic farm in Lebo, and the Carmen Research Station. And so we had a lot of data from our 2017 to 2019 trial where we tested different lines. And so we decided to pull different genotypes from those trials and put them into these trials. And so we made decisions on, you know, what yielded really well, what performed very differently from one environment to the next environment, and we decided to incorporate it into this one. And so if you can pick out your initial and your location, that's probably you. But you can see that we had lots of different spread of geographic locations that we pulled genotypes from. And then the checks we decided to incorporate into the experiment were AAC Brandon, which at the time, and maybe currently, organic farmers were growing quite a bit in Manitoba, at least. It was the leading cultivar being grown. CDC Kernan, we had tested in our past experiments and it performed very well. AC Tradition is the only organically selected and bred registered hard red spring wheat. And then Jake and Zealand both came out of Dr. Dean Spanner's program, which were not necessarily selected under organic conditions, but they were recognized as being suitable for organic production. And then we have Vesper, which performed extremely well in our past experiments in our organic trials. And so we decided to use that as a check as well. So moving on to results and discussion. Some of the results, pictures say 1,000 words. So these are just some pictures from the experiments that we ran across the prairies. And so this was in Carmen, Manitoba. This was in Lebo. I don't know if you can tell, but this was an extremely dry year. This was in 2021. And if you were in Manitoba in 2021, I hope I don't bring back any traumatic experiences. This is Oxbow, Saskatchewan. This is another one, Oxbow. This was Lebo last year. We had a very good year last year. And this is the trial in Edmonton, Alberta. So now we've, tested all of these lines across all these different locations. And so now we have lots of yield data, an overwhelming amount of yield data. And so if I was to, plot all the bar graphs of all the different genotypes across all these different environments, this is what we come up with. And so our yield range across the different environments was between 10 and 65 bushels an acre. These numbers here mean that we did have, we ran the numbers through a statistical program, and we did have genotypic differences. We had massive environmental differences, and we did have that crossover interaction that I was talking about, that genotype by environment interaction. And so just to give you an idea of what we saw, in Carmen 2021 and Lebo 2021, we had lower yields due to drought. In Lebo 2020, we had extremely low soil phosphorus. We had about 3 ppm of Olsen P. In Oxbow 2022, we had heavy wild oat populations. In Roblin 2021, we had heavy alfalfa competition because the alfalfa wasn't tilled in enough, so it became a weed. In Edmonton 2022, we had this really high spike of yield because we had really high nutrient content, no weeds, and 11% organic matter. And so I think that what we saw across these different environments is really indicative of what a lot of organic farmers see. And I'm sure a lot of you can identify with some of the conditions that I experienced in my experiments on your own farm. So we had a really good spread of environmental conditions to test these genotypes under. But at the same time, this bar graph doesn't really tell us very much. What are the questions that we want answered, and how do we observe this data in a different way? How do we capture what did well in these environments, and what did that same genotype do poorly in a stressful environment? Did the genotypes that did poorly in high fertility environments, did they also do poorly in low fertility environments? How can we encapsulate this data and actually pull some information out of it? One way we can do this is by plotting the mean environments. So there's 12 different environments across the x-axis across the horizontal. So this is the average of the environments individually, and you can see that there's 12 of them. And then we have the yields of the individual genotypes. And so this is also very busy and not very informative. And so we want to kind of take it another step. One way to look at yield stability is to compare the different genotypes with how they performed in really stressful environments and how they performed in not so stressful environments. And so we can look at the slope. And so when our slope is very angled, we will have a higher number, which means that it is pretty unstable. It did poorly in poor environments, and it did really well in higher fertility environments or high yield potential environments. And so that gives you an idea that it is a little bit unstable. Whereas if you have a horizontal slope, you have a low number, and that gives you an idea that this genotype is stable. It performs similarly in low environments and low fertility environments, and it performs similarly in high fertility environments. And so what would be wonderful is if we could find a genotype that does this, where it has high yield potential and it doesn't matter where you grow it, it's still going to yield really, really well. Well, this rarely ever happens, and especially in organic environments where we have weeds, fertility differences, variability from farm to farm, variability from field to field. This is something we rarely see, but at the same time, we still want to identify the genotypes that were more adaptable to like many different environmental conditions. So the way we do this is we take the slope number that I show you here, and we can plot it against the average yield. And then we can start to pull some data out of this experiment. So on the bottom axes, you've got the average yield. And then this black line here is the average yield across all of the genotypes. And so our average yield was about 35 bushels per acre across all the genotypes and across all of the environments. And then on the y-axis, we have the regression coefficient. And the black bar that runs along here is known as the average stability. And so one represents, with every incremental benefit of a better environment, the genotype responded equally to that better environment. So we have 4 quadrants. We have a low yield, low stability quadrant, which is down here. So you can see that the yield is low and the regression coefficient is low. And so these genotypes did not respond to better environments as much as others. We also have, just to point out here, this dotted line here and here is the standard error of stability and the standard error of yield. So anything that falls outside of that standard error line represents that it was outside of the average, outside of the normal. So in the upper left quadrant, we have low yield, low stability, and you often don't see a lot of varieties that fall in this quadrant. We have high yield, low stability in this upper quadrant. So that means that these genotypes responded more to high fertility environments, but they also didn't do as well in low fertility environments or low yield potential environments. And then we have the bottom right corner, which is high yield and also high stability, which is this quadrant right here. And so, if you can pick out your initial, if you recognize yourself, you pick out your initial, you can kind of see where you fell. But the letters after each cultivar name indicate statistical differences. So for example, if it has an H after the name, that means that these two yielded the same. If it has F after the name, these two yield the same. If you have an ABC, they yielded statistically similar to each other. You can see there's quite a gradient between the different lines across these different environments. And this kind of curve where you have low yield, low stability, high yield, or low yield, high stability, high yield, low stability is pretty normal. This is often what you see across genotypes in many different breeding programs. Some of the results that I want you to pull out of this that I noticed, you can see that I've got different colors going on here, is that we have 2 lines that didn't do as well across the different environments. And so one of them is Jake, which is a registered variety, and one of them is PWA 10B. The majority of the genotypes, the PPB lines, actually fell in the average stability. And so you can see up here that the registered varieties that we put into the experiment actually also fell in this average yields quadrant. And then we have kind of our little bit of our winners up in the top quadrant, which is BJ13GW, BL43CTM, Vesper, and BL34SW. And then this yellow one I like to call is the golden unicorn. This one yielded just as well as the other high yielding genotypes, but it was a little bit more stable. It was more stable than those high yielding genotypes. And one thing I wanted to point out was that, you know, we have some genotypes in here that are have the same parental line. So BL34 is the same exact, has the same exact parents, but they were selected by two different farmers, and that they were able to change the population dramatically, even though the parents were different. This was already established in our 2018 paper, but we had this opportunity to kind of point this out. And I thought that was pretty interesting to show that farmers actually did influence the population quite a bit. So some of the results in discussion, we established that genotypic variability exists. This isn't in the literature at all in terms of the geographic scope that we tested the lines and the environments that we tested them under. So that's really exciting. The Czechs, CDC Kernan, Brandon, Zealand were low average yield and average stability. And the poor performers, Jake and PWA10BLD, Jake is more suited more towards like very short season environments like Northern Alberta, which is where Dean Spinner's program is, servicing. And PWA10BLD was selected in Quebec, so that may have impacted the adaptation capability of testing it under prairie conditions. However, interestingly, BJ13GW was also selected in the eastern portion of the country, and it was one of the highest yielding varieties. And so, there seems to be different adaptation capabilities between lines. But yeah, BL43CTM and BL34SW and Vesper were all high yield and high stability, or low stability, sorry. And then BL23AS was our golden unicorn with high yield and better stability than any other high yielding genotype. So in terms of, if we were to put something forward for registration in terms of the data that we have, BL23AS may be a very good candidate. However, BJ13, BL43CTM, and BL34SW may also be very good candidates because they seem to react to better environments, and it may react to conventional conditions more so than the other lines. I'm not sure. So some of the takeaways, this is a valuable proof of concept for others that are interested in participatory plant breeding programs, that farmers can generate genetic diversity and provide high yielding genotypes compared to checks, compared to what's out there right now, especially for organic conditions. And this data is also useful for organic farmers because that high yield, low stability is not necessarily a bad thing. If you've prepped your land for high fertility, low yield, low weeds, and you want, bang for your buck of the effort you put in, then you may want an unstable genotype that's able to respond to higher yield potential. However, if you have a very challenging area on your farm, which I know all of you probably can pinpoint right now where that is, you might want something that's a little bit more stable with good yield potential, but is able to adapt a little bit better than those other high yielding genotypes. And this really drives home that organic farmers are not growing cultivars that are best suited to their production system if they're only relying on modern varieties. Some of the questions that I can think of out of this experiment is, what is driving yield stability? I would have loved to have taken a lot more measurements across the different environments in terms of like biomass at different times, or numbers of seeds per spike, and different physiological measurements, but I wasn't able to do that. But I am trying to input some of this data into different regression models to see if there is something that we can pull out of, like what was really driving that yield stability and performance. And, you know, how did certain environmental characteristics dictate which genotypes did best where? And so there's some biplot analyses that I can do where I can look at, you know, these 3 environments clustered in one section, and this cultivar did really well here. Why is that? And, you know, these 4 different environments clustered in one section, and this genotype did well at those 3 or 4, and why is that? And also, what is the impact of environmental qualities of organic test sites and genotypic expression? We see a lot of times when we're doing these testing trials and the whole site has very low yield, we often don't see genotypic expression and differences between genotypes. And there isn't a lot of information and data in the literature that speaks to this challenge in organic plant breeding. That, you know, we want sites and test trials that actually pick up these genotypic differences. And so where is the line between you know, a stressful environment that we can really test drive these genotypes, but also not so stressful that we don't see any differences. Yeah, so those are some of the questions that I've pulled out of this experiment. And I just wanted to thank all of you guys that I could not do this work without all of the hard work that you have all taken out of your busy time and schedules. And also special thanks to the Christian family who hosted my experiments this year, or this past 3 years. So that's it. I'm open to questions.
Awesome. Thank you so much, Michelle.
Yeah.
I have one question in the chat, and maybe while people feel free to type your questions in the chat and I can read them out loud, or after I can, you can unmute yourself. But this first question comes from Naveen. While assessing the performance of these cultivars, was there any common factor across the locations, like weed pressure or fertility, which has major impact on performance?
Yeah, so one thing I wish I did was to take very specific weed population measurements, but at the same time, you know, you have contracts with different cooperators, and so they don't necessarily have the time to you know, gather weeds. And honestly, it wasn't even something that I was going to be testing for. I was really just tasting these genotypes for a test drive. And so it wasn't until I started realizing the power of GGG biplots that I was like, oh, I should have been a better job of characterizing the sites. However, I do have all the soil tests, and I do have all the precipitation and climatic records. And so that's what I've been using to try to characterize the sites. In terms of similarities between, we had such divergent environments that I can't say that there was necessarily lots of different similarities between them. You know, I could say that Lebo and Carmen, they both experienced drought in the same year, but they are completely different soils. Lebo is a very heavy clay, so like this picture right here is actually in Lebo, and Carmen research is kind of a sandy loam. And so, you know, how those 2 soils react to drought is very different. And so we didn't see a lot of differences. And a lot of times, like when I had low soil phosphorus or drought, we didn't have a lot of weeds. And so weed competition was not the issue. But we did have wild oat challenges at Oxbow and alfalfa challenges in Roblin. But when I did the GGG biplot analysis, those 2 didn't really cluster together. So there's definitely something else going on.
Are there any other questions or comments or things that stuck out from what Michelle shared? Yes.
Go back to this.
I have one question here. Can I go ahead?
Mm-hmm.
Yeah, so I don't know whether I have picked up correctly or missed one of the points that I want to clarify from Michelle. Michelle, in this graph, regression coefficient versus yield, I can still see there is a linear relationship on the spots. Is there anything that we can pull out from that kind of relationship?
Like in terms of how, you know, we've got this line that goes like this.
Yeah, exactly. Yeah.
Yeah, it's it gives us an indication that the genotypes that are slightly lower yielding are a lot more stable. So they don't necessarily respond to better environments. They're more, and some papers would say that they are more adapted to poor environments,
Okay.
right? But
Yeah.
then for, from my point of view, farmers need to make money. We don't really, like they need to make money, they need to have good yields. And so do we really want lines that are poor yielding, no matter where you grow them? We want lines that are high yielding and also stable. And so a lot of times, these stability studies, they do follow this linear relationship. And the key from my standpoint was to find lines that kind of fell into this quadrant.
Okay.
This is the key, that you want something that is more stable and also high yielding, because it's more adaptable to many different organic environments.
Yeah. Yeah, sounds good. Perfect. Thank you.
Yeah.
I see Dwayne has his hand up. Would you like to ask a question?
Yes, I have just a couple of comments. One is that I hate the word unstable. I think responsive is a better way because
Responsive,
it's on it. And then when you give something what they would like, they respond and do much better. And if you don't give them what they need, they respond to that too. I have used GGE biplots extensively and looking at environmental effects on varieties. We did an analysis in Australia looking at quality, specifically protein. And we broke it down to rainfall and temperature and nutrients. And we did it month by month by month. And there, since they grow most crops vegetatively during the winter and they flower and reproduce in the spring and early summer, we found that the rainfall in June had a tremendous impact because that's what got things started or not. And the temperature in September and October, the cooler the better, because that meant that it didn't dry up too fast. Those were the biggest factors in protein in Australian bread wheat. And it explained a fair amount of the variability. So Michelle, when you're working with GGE, you can learn an awful lot from it. Especially if you tease out the environmental conditions associated with different locations.
So I don't know if you're familiar, but I used partial least squares regression because I did have some anthesis biomass and mature biomass and things like that from different environments. So I took 7 of those environments and I inputted yield across all these different, the climatic and nutrients and the soil test and everything. I got a pretty good model, explained 95% of the variability, and the important climatic dependent variables that came out was precipitation at anthesis and also growing degree days. So I saw the exact same thing because, like, especially in 2021, we not only had drought, but it was incredibly hot. We had some of the record above 30 degrees Celsius days we experienced in 2021. So that we showed through growing degree days in that PLS model that had a significant negative effect on yield. So yeah, I'm hoping to incorporate that into the paper as well.
Yeah, when you have enough data and can put it in and then get rid of everything that doesn't mean anything, then you can learn something. But it's really hard to separate drought and heat stress because they're often going together and they're not good.
Yeah, but we didn't find that, so I tracked the precipitation between seeding and stem elongation anthesis, anthesis to maturity, and seeding to stem elongation wasn't a factor. From anthesis to maturity wasn't a factor. It was definitely stem elongation to anthesis was a major contributor. So that was interesting to see.
Yeah, anthesis is an extremely stressful time in a plant. And if you don't get pollination, then that immediately reduces your yield component. So in almost every crop, that week or two around anthesis makes a phenomenal difference in the final outcome. but good luck on controlling the environment, right?
Yeah.
Awesome, thanks, Dwayne. Maybe we'll do one last question from Ward, and then feel free to save your questions for after. We're going to have Martin's presentation, so you'll have lots of time to ask Michelle more questions afterwards. Yeah, go ahead, Ward.
Honestly, I could wait as well, because Martin might want to chime into it, or it may change my question. But essentially, the question for Michelle is regarding the wheat that you had sent me back that I'm propagating on my farm. That's only really partway through. It was at year 6, and this is what I'm propagating now with no further selections, with the intent to get it to field scale to sell into my farm direct markets. Part of the philosophy that I've been embracing with you using this as my primary wheat, hard red spring wheat production is that it, with more genetic variability or diversity, I theoretically would have some built-in protection to get some crop in the face of extreme weather conditions with climate that are going to come with climate change. And so when I look at the green circle, like the one that I'm growing is the BL39A, that's kind of in the top right-hand side of the circle in that cluster on your page that's showing right now. And you know, would I be better to forsake this and move to something that was, you know, in the green circle in the top right? Or in your opinion, does that, if it's more stable, does it also then present more risk in the event of an extreme weather season, like extremely wet or extremely dry? It'll be difficult for you to test both, you know, because it depends on the season. Like you'll do an average, but I'm looking for getting some crop every year, not necessarily the highest crop every year.
Right. Yeah. I would say, so this is the thing, is that we tested them, we didn't necessarily, it'd be wonderful if we could test your lines on your farm over multiple years, right? But we just can't do that. And for this kind of stability analysis, breeders say that you need about 10 site years of data to really capture, an idea of yield stability. And, you stopped at the F6. And so each of those plants are homozygous, meaning that those plants are not going to change because wheat is a self-pollinating crop at the F6. But your plants are still heterogeneous. You're still working with a population where individual plants within your population are different depending on your selection pressure. So if you selected, through the 3 years, you selected 3 spikes, then your selection pressure is quite high and you're going to end up with a pretty homogeneous population. Like what breeders do, like breeders would take 1 spike and they'd plant a head row, and then they would evaluate that head row and then take that 1 head row and move it on to the following breeding, process. However, in PPB and what you did, you selected 300 or 500. And so you selected the best 300 or 500 over the course of 2 years. And so your population is still heterogeneous. You still have different plants within your populations. And so, and you've continued to grow it on your own farm, and through natural selection, it's going to keep on changing and growing with your own farm. Whereas BL39AWM up here that you had mentioned, we've been growing this on our research trials. Like this is saved seed from our research trials. And so, what would be really interesting is if we could test the performance of your saved seed and our saved seed and see how these different populations, there's just so many research questions you can pull out of this. But I would say that even though it is in this upper right quadrant, and it is like high yielding, you can see the letters after this population is A, B, C, D, F, like it's not significantly different from your higher yielding. This average stability right here, this, this dotted lines here, the standard errors, like it's still good stability. It's not a racehorse. And so I would still be confident that when you grow that population, you're still going to get something even in more difficult years. It just may be more responsive, as Dwayne said, to better environments than some other lines. You know, you can throw like PWA 10B, LD, Jake, you can throw as much nutrients as you possibly can. It's still going to yield poorly, which is, you know, what this graph is, what this is telling us. And so I still I'm very confident that you have a good line and that you're still going to get something in stressful environments. And if, you know, if anybody wants information on their own line and the yield data from their own line across different environments, feel free to, you know, shoot me an email, because I'd be more than happy to share this data with you. For example, Ward, if you wanted how your line did in drought, in our drought conditions, and how your line did in, you know, heavy wild oat conditions, you know, very happy to share that.
Perfect. Thank you, Michelle.
Yeah, thank you. That was a great question. Maybe just in the interest of time, I will pass it over to Martin to continue the presentation and discussion.
Okay, thank you very much. Good morning, good afternoon, everyone. It's really great to be part of this conversation. This is a really great community of people. Hopefully you can see that slide. It should say Glenlee long-term rotation.
Yeah, that's good.
Okay, great. So what I wanted to do is really build on what, you know, Michelle's work has shown us, and that some varieties are better suited to organic production. And so what the focus of other research is to try to bring those into production systems. And one of the places we do that is at Glenlee, and starting in 2021, we've in the organic plots at Glenlee, we've actually put the PPB lines into the study. Just a little introduction. In terms of long-term organic studies, there's about 35 or so around the world. Glenlee is the only one in Canada, and it's going to be entering its 32nd year this year. So this is a picture of an aerial shot of the long-term study just south of Winnipeg. And this is actually on a Métis River lot, and there's two basic rotations. One is just grains, wheat, flax, oats, soybean in the conventional or green manure in the organic, and then 2 years of alfalfa for hay, and then wheat, flax, again, both in the conventional and the organic. And we split the plots a little bit to add some manure or not add manure. So these are the main rotations. We also have the prairie plots in there, which is a really nice benchmark. So that's a little bit about Glenlee. We've measured things. We've done many measurements here. We know these soils quite well. We know how they react. For example, this shows nitrous oxide emissions over 2 years in the conventional and the organic wheat. And we have learned that the organic wheat system at Glenlee is emitting less in nitrous oxide than the conventional, and even less per bushel of production. And just in giving you a little more information about Glenlee, the soils, and I think it is important for us to understand what these organic soils are like, because that helps to maybe explain why varieties, lines are performing well in them, and why they might not be. So this is a lot of information, and it's kind of new, and it's really exciting. So let me just show you the cropping systems. Just going to walk through this. So we've got the prairie. Then we've got the C's are the conventional rotation. So we've got the conventional grain only rotation. Then we've got the organic grain only rotation, the forage grain rotation under conventional, and then 2 of them under organic. But this forage grain rotation receives manure, but a low level of manure. We only add manure enough to satisfy the phosphorus requirement, or to make sure the plants aren't phosphorus deficient. And then when we look at, soil carbon, which is whatever we're all talking about now, we actually broke it into two groups. We have total soil carbon, and then we have microbial biomass carbon, which is really the gold standard of soil health. And we learned that the grain only rotation doesn't perform that well when we do it organically over the last 3 decades, because we basically haven't put enough organic matter in. We've changed that now. We've altered this system, adding more cover crops, and we're trying to reverse it. But then real high performing system is this forage grain organic system. Whether we add manure or not, it performs better than all the other systems, including the conventional forage grain system. So there's something about that organic management that's allowed us to have a better soil active carbon pool. I don't want to spend a lot of time on all these other measurements. Let me just point out the water stable aggregates. This is a very important soil health measure, the aggregate stability. And here we find the prairie has the highest aggregate stability, no big surprise there. But the forage grain with manure has a statistically similar aggregate stability as the prairie, which is almost shocking because we till this rotation, we don't till the prairie. Very, very interesting. And then some of the others fall in between here. And then these 3 parameters are enzymes. This enzyme breaks down carbon, this enzyme makes phosphorus available, and this enzyme makes sulfur available. And we see a lot higher enzyme activity in the organic systems generally, and especially in so if we look here at aryl sulfatase, it's higher in the organic grain only and much higher in the forage grain, and significantly higher where we add the manure. And then finally, the mycorrhizal colonization, the highest level of these beneficial fungi, usually they're beneficial, is in the prairie, no surprise there. And then among the farming systems, they're almost always higher in the organic version. So for here, the organic grain only, almost 50% of the plants are infected versus 28 in the conventional. 32 in the conventional. And I've got a new student, Letitia Makungu. She's continuing to look at mycorrhiza at Glenlee, and this is data from 2022. This is flax, and here we are in the conventional forage grain rotation, the organic forage grain rotation with no manure and with manure. And these are both higher than the conventional. So really, what I did is just very quickly paint a picture of how active the soils are in these systems. And so what we've learned is there's some organic systems that are not doing particularly well and some that are doing very well. One other interesting piece of data that we've been monitoring in Glenlee for probably 12, 14 years now is that the pH of our organic systems are remaining stable. The pH of our conventional system is slowly tracking down to more acid. And this is something that was written up in the Western Producer a while back. And it's also something that the other long-term organic conventional studies have documented. And that, that we don't want the pH down at 5.5. We want the pH to be neutral, because that's where soil health is much better. So that is a little bit on the soil background. Now let's take a look at the PPB varieties. So starting in 2021, we made the decision to grow the conventional variety in the conventional plots, and we have been growing AAC Brandon. And then we grow one of the PPB lines in the organic system. And this is a picture during the 2021 drought. It was a devastating drought, and the lines look very different. We grew the line called HRE in here. And if we look at the yields, under these really stressful conditions. They're shown here. The black bar is the total biomass that they produced, and the hatched bar is the grain yield. And what we found is that where we grew wheat after alfalfa, especially under organic, we had very low yields because we had no water. We did a little better in the perennial conventional because we use no-till here. But that's probably not the only or other reason. Perhaps the other reason is we're fertilizing these rotations, whereas here we're relying on mineralization of nitrogen, which doesn't happen when you have no water. But over here in our conventional grain-only rotation, I mean, the yields are all very low, but the two organic yields were exactly the same. And so these are PPB varieties. This is a conventional variety. There's some variation in these rotations, which I'm not going to take the time to explain. So this was kind of interesting. Sarah Wilcott, who runs this experiment, was going, she was a little surprised. And, we were quite excited. So we did the same thing again in 2022. We used this variety called HRE. It has a variety name. We actually, I think we're going to switch from HRE to the Terry Muro variety, the TM, which Michelle talked about, because I think it's got higher yield. It's more responsive. But in 2022, we switched our conventional variety to Starbuck, and we don't willy-nilly switch varieties, but Starbuck has got better fusarium tolerance. It's really an improvement over Brandon, and we'll probably grow it for quite a while. And if we look at the conditions, the yields in 2022, we can see that, again, under conventional production, that's our organic in the rotation that's had more cover crops. That's our conventional. The yields are almost identical. And then if we look at our perennial forage grain organic with the manure added, it's actually yielding the same as the conventional, and the same as the conventional in this better rotation. So I think what we're starting to see is I'm really happy that we put the PPB lines into the Glenley rotation, because I think if we would have put Starbuck or AAC Brandon into the organic, we would have disadvantaged it. Now, we do have some challenges in organic, and that is seed-borne diseases. We are suffering from some loose smut in our wheat, and cleaning that up is challenging. We have to be really careful of the seed source because we had a little bit of smut in the HRE, and I just want to point that out. Now, one of the other things we're doing in the long-term plots at Glenley is changing our management to improve soil health even more. So we've started going to a very minimal till system in the organic using the wide blade cultivator. Last fall was the first time we did this as the fall tillage to kill thistle. And, so our organic system is evolving. And with that, I think our adaptability of our PPB lines will also evolve. So what are the next steps in the PPB program? First of all, Murray Jowett is a master's student in the Natural Resources Institute at the University of Manitoba, and he did a value chain study to look at how these lines could be moved into the market by farmers. It was very interesting. That thesis should be available online. But I think what we want to do is move some of these more promising grower lines, including wards over here, onto farms. So this one here, for example, comes out of southwestern. This is Stuart Wells's. This is Allison Squires. This is Terry Muro. This is the one that I want to start growing at Glenley. I think bringing these onto farms, perhaps farms in the area where they were selected, would be very interesting. And the other thing we're continuously doing is making these PPB lines available to plant breeders for their future crosses. And there could be other development steps that we take. For example, Abir talked about the possibility of some registration. I want to say just a little bit about the oats, because we also have the PPB oat breeding program. And these are the results from the Borg experiments. On the left is 2020, on the right is 2021, and we don't have the 2022 data yet. And here you can see the Czech varieties, Morgan Summit goes down the list here, and their yields and their rankings compared to the PPB lines. The numbering system is different for the 2 years. What we can see is the PPB lines have not performed as well in the oat program as against the Czechs as they did in the wheat program. Now there are some promising lines from the PPB oats. This one here continues to be in the Borg trial. So that's what we've learned. And the Borg trials, we have 2 locations in each of the 3 prairie provinces every year. So it's a fairly comprehensive organic screening program. And this is the Borg trial at Lebo in 2022. And you can see it's a beautiful, beautiful hoot crop. The one thing I do want to point out that's different in the oat industry is that we've had more directed breeding for organic variety. And Jennifer Mitchell-Fetch released Kongsor, AAC Kongsor, and it ranks fairly high under organic. I mean, Morgan is still the top ranker, but it's got very poor disease resistance. And so that's a challenge, especially in the eastern prairies. So what we've done is looked at, and this is just my last few slides. What we've done is looked at Kongsor compared to some of the other oat varieties in terms of how they intercrop with legumes. So it's been interesting work that Heather Flood did working with us. And I'm just going to quickly show you the data here. You've got on the top left is the grain yield, the Kongsor by itself, Dancer by itself, Morgan by itself. And here's a PPB line, one of the better performers. Comes from northern Alberta. So that's the yield on its own. And then the orange bar is the yield when it's intercropped with peas. And the dark bar is when it's intercropped with vetch, as a hairy vetch as a cover crop. So, that data kind of speaks for itself. The next location here, this is the same experiment, but at Glenlee at a different location. And we can see that the peas are dropping the oat yields quite a bit, but we are getting a pea yield from there. The vetch, not so much. I'm going to go back to Carmen and just point out the bottom left here is the effect of the intercropping on seed size. And one of the things that really stands out to me here is that Kongsor has consistently large seed. And it does have a higher percentage of plumps generally, and that's one of its features, growing it organically. So here, the intercrops, it has the highest seed weight generally, and was not bothered too much by the intercrops here at Carmen. If we go to Glenlee, the next site, you can see again, really none of the varieties were bothered by the cover crop. In terms of the plumpness, but Kongsor again had very good plumpness at that site. So the idea of breeding for intercropping is something that's interesting to a lot of people, and I think it's worth spending more time as plant breeders. So what are the next steps in the PPB oat program? Well, all PPB lines continue to be evaluated by Dr. Kirby Nielsen of Ag Canada at Brandon. And again, I think moving more promising farmer selected lines onto farms might be a possibility. Again, making the lines available to plant breeders for future crosses. And, one of the things under other, I think, is to think about selecting oat lines under an intercropping scenario or under a cover cropping scenario. So thanks for your attention. On behalf of Michelle and I'd like to thank you for participating. There's Michelle and I standing in front of her 2022, one of her sites. I'd like to thank the participating farmers. It's a lot of work, what you did. And I think when we started this program, we didn't recognize really how much effort it was for the farmers. And I just, want to thank you so much. And also the students and our funders. And I think, Tierra, I'll turn it back to you.
Oh, thank you so much, Martin. It's really exciting to see the PPB varieties integrating into intercropping and the Glen Lee trials. That's really fantastic. Maybe while people are thinking of their questions, either typing or about to ask in the verbally, we do have a question from Naveen. Naveen asks, even if the oat variety doesn't perform well as compared to Czech varieties, does it have better or comparable protein content or quality aspects to Czechs, or relatively in comparison to wheat PPB varieties performance?
Yeah, because there were good parents used for the oat crosses, the answer is yes, the quality is good. And the beta glucan is all those things are being looked at because they're in the Borg trials. And so Kirby Nielsen has that data. I have it as well, but you could talk to Kirby. They're really at Brandon. I'm sure he'd be happy to talk to you. The one thing we do know is that Kongsor and Orvina have very good beta glucan, but the industry only wants a certain percentage.
I do have another question related to oats. Like when we are talking about intercropping in oats, so is it possible that we can grow oats again the next year, like within an intercrop?
Yeah, that's the question that always comes up with intercropping, right? I don't think it's a good idea to grow the same crop 2 years in a row.
Because of volunteers or just?
Well, it is taking a risk to do that. But we can all get away with it once or twice. Yeah.
Okay, thank you.
Any other questions or comments or things that stuck out from Martin or Michelle's presentations?
Well, one of the questions in the chat, if it's okay, Stuart asked about, is anyone looking for a blend of the evolutionary blend? He has it available. So that's great. Thanks, Stuart.
So can I ask another question related to organic no-till rotation?
Sure.
It is not related to this, yeah. So I was wondering, like when we are creating a mulch of a cover crop, so is it a good idea to do spiking of that mulch to have better aeration and water infiltration in the spring?
I don't think that's necessary at all. What that will do, I think, is move some of the mulch aside and allow weeds to come up.
Weeds to grow. Yeah, okay.
I think unless you have a mulch of 6 to 7 tons per hectare of dry matter on the surface, you're not going to suppress weeds, and especially wild oats. Wild oats are not suppressed well by mulches, and if you read our literature, that mulch system worked well unless you had wild oats or a lot of wild oats.
Okay, and doesn't it delay the planting in the spring because the soil is still like cool or not relatively warm enough?
Well,
the mulch.
One thing about no-till is that you can seed very shallow, and so yes, the soil is cooler. There's no question, but you can, you don't have to put the seed quite as deep, and that can sometimes compensate for some of it. But we've compared. One of my grad students, Sam Curtis, compared organic no-till into a vetch barley mulch, no-tilled versus a conventional wheat. With conventional no-till after either canola or soybean with herbicides, and And the organic wheat yielded exactly the same.
Yeah.
That if there's legume in the mulch, there's a lot of nitrogen. It's a great, it's a great environment. But
Mhm.
you know, that's why it's important that you do some research to find out what works in your area, because you
Yeah.
are in a shorter season area,
Yeah.
and the soil temperature is a factor for sure.
Yeah. Yeah. Thank you.
Maybe we have time for just one more question or comment.
It's Ian here.
Yeah.
Great presentations, really fascinating work, both Michelle and Martin. Is there merit to, because we have difficulty marketing non-registered varieties into market for organic grains. It is possible to do that on a small scale to direct end users, but a lot of our customers are not small scale. Is there merit to, we grow commercial varieties for that reason. Is there merit to further selection by farmers on their own farm with the sort of the top varieties that we consider that we should use on our farm? Is there merit to actually selecting those and trying to improve them through the selection process we used with the farmer selected varieties? Just wondering what your thoughts are on that, whether that's
I'm going to let
pie in the
Dwayne
sky.
I'm going to let Dwayne or Michelle answer that question.
Okay.
Okay, having been on some of those committees with CFIA, it's complicated. The problem with taking a farmer selected variety through the registration system is time and expense.
But that's not his question, Dwayne. Let me just interrupt you. That's not his question. He's wondering if he could select from his AAC Brandon on his farm.
Depends on whether there are variety rights associated with that, plant breeders' rights. It may restrict what you can do with a specific variety. The best thing to do is contact the breeder and see what they would suggest, because some are pure lines and there's very little room for selection unless it's a new mutation, and most of those are not positive. If it's a bulk of somewhat heterogeneous material, there may be an opportunity there, and it may be possible to work directly with the breeder and make some real progress and come up with something that is very worthwhile. But a lot depends on the variety itself and the origin. It won't be easy no matter what.
Okay, just a supplementary question. We use a variety, it's a varietal blend, and it would be typically, well, it's a varietal blend to resist wheat midge. So it's a blend, has a refuge in it. That would probably not be a good candidate to save, but if we select the best seeds and the best heads for years, and we maintain the variety, and we're allowed to save our own seed into the future and call it the same variety. Prior to plant breeding rights, that was all possible. As you mentioned, it depends on the origins and the parentage of the variety. Is it theoretically possible to improve variety, given the right conditions and the right selection process?
I guess I can jump in here again. Absolutely. However, if they are a pure line selection, you probably won't be able to improve them very much. If they are essentially a landrace, there may be considerable scope for it, particularly for adaptation, or if you have an environment where you can select for disease resistance or stress resistance, it may be possible. And land races are basically what breeders produce by making a cross and growing out the early generation segregating material. And this is basically what was released to the farmer plant breeders, were a raw land race. And so there was lots of opportunity to select for selection. And it may be possible that some of the public breeders may make some of their early generation material available to farmer selectors. It may depend on the institution, it may depend on the breeder, but it may be possible to get material that has a tremendous potential for selection by getting early generation, highly heterogeneous material and growing it on your farm, selecting it with your eyes for your environment. There is a lot of potential there. That's what our ancestors did for thousands and thousands of years, and that's what got us to where we are now. And that's what plant breeders do.
Right. Could I just jump in here? I think the challenge is that Ian can't sell that to the elevators, like his markets. And so, like, I think, and Dwayne, you could correct me, but in terms of self-pollinating crops, especially when you're dealing with something that's been registered, and the majority of wheat lines that are registered are usually out of pure line selection. You know, because it's self-pollinating, it doesn't really outcross and genetically mix with its neighbors so that you get like a unique line very much. I would say it's like 95%. It's like 2 to 5% of that population would outcross. And so, you know, over time, you might see incremental changes, but in terms of, you know, when you're looking at 2 to 5% of your whole population outcrossing, you know, that would be, I could see that as being beneficial over a very long period of time. And that would be a very interesting research project, you know, thinking from a research standpoint. I know they've done like evolutionary plant breeding where they sent F3s, like what we did with PPB, and farmers didn't do active selection. They just did a natural selection process, and they grew out those lines over 10 years. And they did see differences between farmers and farms, you know, naturally selecting over time. But in terms of selecting straight from your registered variety, it also calls into question the seed quality. If you're constantly saving seed on your own farm, you know, the seed-borne diseases that come out of it. And also, you know, if you have a really tough year, like a really droughty year, your seed quality is also going to go down. And so these are just things which I'm sure you know, to consider. But I do think it would make an incremental difference, but self-pollinating crops usually don't outcross very much.
Can I add a comment there? And Ebir was also on Murray Jowett's thesis committee. He was quite involved, very helpful. But what Murray learned is that a lot of the buyers, talked about, is that what the buyers want from large commercial, for example, wheat growers, like many of you on the call, they want quality. That's why they want a particular variety. They want the quality. And so it may, I don't think we're there yet, but it may come to pass that if a PPB line had good quality and a good supply because of good yield, then a company might say, we're going to just direct buy it based on its quality, not on its genotype. And that may happen in the future. The other thing that may make it happen faster is something that I don't want to see happen, but if we get more restrictive regulations around wheat, or if we start having GMO wheat, organic producers are going to be in a very different boat in terms of the varieties that they will have available to them, but we're not there yet, and hopefully we don't go there. But those are some other thoughts.
Thank you so much, everyone. Time always seems to escape me whenever we're having great discussions here today. So I think I'm going to have to wrap things up, but a huge thank you to Martin and Michelle for being here. It's so incredibly great and useful to have this kind of data available and out there. So thank you. And thank you to all the farmers who participated in this data, contributed your time and being here for this great discussion.